Hello, good evening all. It's JNV Gandhinagar and JNV Silvassa. Okay, good evening all. Welcome back to the class. Okay, so today let us study some more topics of the human reproduction. Okay, before going to the class, I would like to ask you to fill the poll because whether my voice is audible clearly and is the blackboard visible clearly. Please answer in the poll so that I can understand if any changes are required. Am I audible? Yes, that's great. Many people had to join. Okay, I'm ending the poll. Thank you for the confirmation. Okay. So what we are discussing today or in today's class is that about a hormonal control and a gametogenesis. So if you all can remember yesterday when I was explaining the female reproductive system, I told you there are primary sex organs, right? In the both male and female reproductive systems, we can see the primary sex organs. What is the main importance of this primary sex organs? Can anyone write the chat box? Or can anyone answer in the chat box? What are the importance of these primary sex organs? Anyone in the class? Okay, at least try to answer yourself. I know many people, you are sitting apart from the system. Okay, fine. This primary sex organs will produce the gametes, right? Yes. So in the male reproductive system, if we are talking, there is a testis which produces a sperm, and in the female reproductive system, there is an ovary which produces the egg. But yes, we have studied produces a sperm and it produces an egg. But how it is going to be produced, how the gamete will be synthesized, that is called as a gametogenesis. So gameto, which means the gamete, genesis is the synthesis. So here we are going to study about synthesis of a gamete in the humans. There are two types of gametes will be produced in the humans. One is the male gamete, that is a sperm, and one more is a female gamete, that is called as a egg. And along with this gametogenesis, we will be also studying, yes, we will be also studying the hormonal control, right? So this hormone, just a minute, give me a minute, please. Yeah, sorry, let us continue. So yes, this is what we are going to study, that is the gametogenesis and how does the hormones will be controlled for this gametogenesis. As I said already, like yesterday while we were discussing, right, are you all able to, if you are able to recollect, I was telling you, like, yes, in the ovary the egg will be produced like a female gamete. Along with that, some ovarian hormones will be produced. What are those ovarian hormones? Can anybody mention? That is the estrogen and progesterone, right? Yes. So that is what these hormones will be controlled the gametogenesis. Like the same way in the androgen will be controlled the gametogenesis in the males. That will study. Okay. So is it clear what we are going to study in today's class? That is a hormonal control and yeah. We are going to study about a hormonal control and the gametogenesis. Is it clear? Are we good to go? Are we good to move? Please answers in the chat box. Okay. Yes. So what are the hormones and what are the glands? This is the question. Is anybody, can you please answer? Can anyone please answer for this? What are hormones and what are glands? Glands are the one from where we will be. Yes. Glands are the one from where we will be producing, or they will be producing the hormones. Hormones. And hormones are the chemicals which controls our body functions, like how neurons will control our body function. The same way, there are some chemicals which controls our body functions. Those are called as a hormones. Here, yes, as I said, reproduction is also one of the physiological activity, and this physiological activity will be controlled by the hormones called as a sex hormones. Okay, so we'll study. Yes, so in the human reproductive system, as I said, hormonal control, right? So mostly we will be studying, now we'll be studying about how the hormones will control the male reproductive system. Okay. So the first gland we'll be studying about is a hypothalamus. Hypothalamus will release us the secretion. Okay. So hypothalamus will be releasing a secretion, and that will be entering into the anterior pituitary gland. Okay, that will be entering into the anterior pituitary gland, and from there the hormones will go and stimulate our Leydig cells. Where the Leydig cells will be present, or can you tell me the another name of these Leydig cells? Yes, in between the seminiferous tubules, we can see the Leydig cells or the interstitial cells, and these cells will produce a hormone which is called as a testosterone. Testosterone is called as a male sex hormone. Okay, and what is the use of this testosterone, which is going to secrete a sex organs, or sex organs, or the accessory sex organs, accessory glands, all these will be function based on this testosterone. Okay, in the same way, the hormone which is released from the anterior pituitary will also affect on the Sertoli cells, and from the Sertoli cells also will be getting the hormones called those hormones will helpful for the stimulation of the spermatogenesis. Again, the word called as spermatogenesis. As I said already, genesis means a synthesize, and the spermatogenesis or spermiogenesis means the formation of a sperm. Okay, so how it is going to happen, that we will see now. Okay, so spermatogenesis, where it is going to happen? Any guesses where the spermatogenesis is going to happen? The site of spermatogenesis or the place of spermatogenesis is a seminiferous tubules. Seminiferous tubules. And when it starts? At the age of puberty. And what is the function of spermatogenesis or spermiogenesis? Sorry, spermatogenesis, that is the production of sperms. What is the use of sperm? Why the sperms has to be produced? Yes, to continue the next generation, the sperms has to be produced in, or to continue the next generation, or to transfer the genetic material from one generation to the next generation. It is transforming from the male through a sperms to the next generation. Is it clear? Spermatogenesis, where the spermatogenesis will happen. Seminiferous tubules, if you all can able to recall the structure of the testes. In the testes, you can observe the seminiferous tubules. Okay, seminiferous tubules, later they will connect to the retate testes. Do you all remember from retate testes and then so on? Do you all remember that? Yes, very good. Okay, and when it starts? At the age of puberty. Okay, now let us move to the actual spermatogenesis. So spermatogenesis usually offer two steps. One is a spermatocytosis and a spermiogenesis. Spermatogenesis is not a single step, it is a two-step process. One is a spermatocytosis and one more is a spermiogenesis. What do you mean by this spermatocytosis and spermiogenesis? Okay, now let us see. Spermatocytosis, which means the formation of a spermatid from the germ cell. Okay, so don't get confused. I'll tell you, like, let us say if this is the wall of a seminiferous tubule, which is having a flat cells. Okay, these flat cells are called as a germ cells. These called as what? Germ cells. And now these germ cells undergo some changes. I'll tell you what are those some changes. I'll mention each and everything detailedly, but now I'm just explaining you what is the spermatocytosis. What happens means this germ cell will produce a non-motile structure, non-motile haploid structure. Remember, germ cell which is having a 2 sets of chromosome, which means a 23 pairs. Okay, so this diploid germ cell will undergo a process called as spermatocytosis to produce a haploid non-motile structure, which is called as a spermatid. This is what? This is the spermatid. Now this spermatid undergoes some changes, and this spermatid will gives us the actual sperm or the motile and a functional sperm. Spermatid was a non-motile, non-functional. So that undergoes the changes, and that change is called as the spermiogenesis. That change is called as what? A spermiogenesis. And because of that spermiogenesis, then they will be getting sperm, which is a motile and a functional. Is it clear? It is easy. Spermatogenesis, which contains 2 steps, spermatocytosis and a spermiogenesis. Spermatocytosis means the formation of spermatid from the germ cell. Spermiogenesis means formation of a motile functional sperm from the non-motile, non-functional spermatid. Done? Is it clear? I hope this is. Yep. So we'll move to the next. Now what we are doing is we will be studying in detail about a spermatocytosis. I said no, diploid germ cell is becoming a haploid spermatid. How is that possible? Okay, tell me, do you all remember the mitosis meiosis process? Mitosis meiosis, do you all remember? So if we are talking about it, meiosis, which is the reductional cell division, and the cell division, right? So equational cell division means whatever the cell which undergoes a mitosis, it gives it 2 daughter cells, and these 2 daughter cells are having the same number of chromosomes as the parent cell. But in the meiosis, what happens? Meiosis, there is a one cell which is going to divide into daughter cells, and the daughter cells are having a half of the number of chromosomes compared to the parental cell. Understand, this is what called as a meiosis, as the chromosome number. Hope this is clear. If you are having doubts regarding the mitosis meiosis, please use the chat box, ask the question if you have any, then I can explain you. As I said yesterday, if you're not using the chat box, I can assume like, okay, you understood everything. Okay, chalo, let's move. Okay, first step in the spermatogenesis is the spermatocytosis. Spermatocytosis is again divided into 3 steps or 3 stages, we can say. One is a multiplication stage, growth stage, and the maturation stage. What are these multiplication, growth stage, and maturation stage? You don't worry about all these now, okay? But keep in mind, this spermatogenesis will start from a germ cell which is present in the seminiferous tubule. Clear? Okay. Now look at the diagram. Look at the diagram. Let us only study about this. I am not able to get the color. Just a minute. Let me take another color because yellow color will not be visible properly. Yeah, sorry. Okay. So look at the diagram. Here we can observe the primary germ cell. As I said already, we are not talking about all the germ cells which are present here. Let us talk about only a one germ cell. Let us say we have taken this germ cell. Okay. And I said there are two sets of chromosomes, right? Diploid chromosomes. What is going to happen to this germ cell first? In the multiplication division, this germ cell will undergo the mitosis cell division. Mitosis means what? Equational cell division, right? So one, obviously the mitosis, let me write this also. Like in the mitosis, we can get a 2 daughter cells. In the meiosis, we will be getting a 4 daughter cells. Okay. So here, first it undergoes the mitosis. Okay, it undergoes the mitosis. As it undergoes the mitosis, we have got 2 daughter cells and they undergo again mitosis again. So ultimately the daughter cells which we are getting here are called as a spermatogonia. Why the mitosis is happening here? Because if the number of cells are more, then the number of sperms will be also more. Otherwise, what happens? I mean, we all know that sperm count is the very much important for the successful fertilization, because the sperms has to move or they have to cross all the obstacles. For that, one strategy is to increase the number of sperm. So to increase the number of sperm, firstly, what is happening is the germ cell which undergoes the mitotic cell division or mitosis, both are same. Okay. And it is going to give the result called as a spermatogonia. And keep a note on this ploidy. That is, see, first is a 2x, that is a diploid. We can also write as a 2n. Okay, diploid. And then after mitosis also, as it is an equational division, we are getting the diploid spermatogonia. Remember what is the first we are getting here is spermatogonia. And this phase is a multiplication phase. Is it easy? Just the cell is multiplying and getting a multiple cells. From one cell, we are getting a multiple cells. This process is called as a multiplication. This process is called as what? Multiplication. Is it clear to everyone? Okay. Now what happens? All this spermatogonia which undergoes the growth phase. Growth phase where they are going to increase their size. Is it clear? In the growth phase, they will be increasing their size. All the spermatogonia, but for our convenience, we are looking into the only one spermatogonia. But keep in mind, all the spermatogonia will be undergoing the growth phase. Now, what is going to happen? Spermatogonia will undergo growth phase and increases the size of it. This is called as, now this increased growth of spermatogonia, this is called as a primary spermatocyte. These words are very important. Okay, spermatogonia, primary spermatocyte. And when we will be calling spermatogonia, after the first multiplication phase, spermatogonia. Okay, and after the growth phase, we are calling it as a primary spermatocyte. And now, so pay attention. In this, after the primary spermatocyte, of course, no change in the chromosome number, same diploid chromosomes. Now what happens after the growth? The cell enters into the into the maturation phase. The cell enters into the maturation phase. What is going to happen in the maturation phase? That is a meiosis. As we already know, meiosis is a two-step process, meiosis 1 and the meiosis 2, right? I hope you all know. So meiosis 1 and meiosis 2. As I said, how do we get a 4 daughter cells? See here, if there is a 1 cell, first meiosis 1 happens. We are going to get a 2 haploid cells. And from this, again, meiosis 2. And we are getting from each cell 4 cells. So this is how we will be getting the 4 daughter cells from a 1 cell. So as we are talking about only one primary spermatocyte which undergoes the meiosis 1. After undergoing the meiosis 1, it results into the 2 haploid cells. One primary spermatocyte will result the 2 haploid cells. Those are called as a secondary spermatocytes. Keep in mind, till primary spermatocyte, all the cells are diploid. But in the secondary spermatocyte onwards, all the cells are haploid. Is it clear? Only meiosis 1 happened so far. Now is going to happen meiosis 2. In the meiosis 2, this secondary spermatocyte will divide again, or it which undergoes the meiosis 2 and gives the non-motile, non-functional cells called as spermatids. Is it clear? Is it easy? So let us revise. Let us go back. Let us go back. See, there are 2 steps in the spermatogenesis. What are those 2 steps? One is a spermatocytosis. One more is a spermiogenesis. Let us keep a spermiogenesis aside for some time. We are talking about a spermatocytosis. Spermatocytosis means the same thing happened, right? From the germ cell, we got a non-functional, non-motile spermatid we got, which is a haploid. So is it satisfied? Okay. What happened first is a multiplication phase. At the end of multiplication phase, we will be getting a structure called as a spermatogonia. Right? At the end, we'll be getting a spermatogonia. And what is a ploidy? That is diploid. And after the growth stage, I mean, spermatogonia will enter into the growth stage, which is called as the primary spermatocyte. And now this primary spermatocyte will undergo the maturation phase. So maturation phase is both meiosis 1 and meiosis 2. I'm writing as a M1, M2. So meiosis 1, which gives the secondary spermatocyte. And then the secondary spermatocyte undergoes the meiosis 2, which gives a spermatid. You know why I'm getting these extra lines? Okay. So is it clear to everyone? Is it clear, or any doubts you have? Only spermatocytosis, clear. Okay, very good. Jamie Gondia. Thank you. Thank you for confirming. Okay. So now what? Now we have one more step in the spermatogenesis, right? That step is called as a spermiogenesis. See, spermiogenesis is nothing but the spermatids will be converting as a sperm ultimately. That's it. This is called as a spermiogenesis. What is going to happen here in the spermiogenesis means spermatid, which is having all cell organelles as it is, but whenever the spermatid is converting as a sperm, some of the cell organelles will change and ultimately the sperm will attain a functionality and sperm will attain the motility. Is it clear? Yes. We'll see. Now we'll see the structure of a sperm. Then we will understand which structures has been changed and how do the sperm will get energy for the motility, and everything we'll be seeing in the next slide, if this slide is clear to everyone. Yes, it is clear. Very good. So you can see again, if you want one more time, like I know here I completely messed up the slide. Here it is a proper slide. You can see at a puberty, first is a spermatogonia, which undergoes the which stage? You know, it will be great if I have many people active in the chat box, please. Okay. So first one is the germ cell, which undergoes the multiplication phase, which gives us spermatogonia. And now the spermatogonia will undergoes the growth phase, which gives the primary spermatocyte. And after the primary spermatocyte, where the first meiotic division will happen, then we are getting a haploid secondary spermatocyte. Remember, secondary spermatocyte onwards, all the things we are getting till a sperm, everything is a haploid. But before the secondary spermatocyte, it is diploid. Okay. And then second meiotic division, we are getting a spermatids, and now the spermatids will go differentiation, and ultimately we are getting a spermatozoa or sperm. Both can be called, or male gamete, that also you can call. Okay, don't confuse with the names. Spermatozoa, sperm, or the male gamete, anything can be called. Now, yes, these are all we have seen. Now I'll explain you the structure of a sperm. So sperm, which is having a 4 structures: head, neck, middle piece, and a tail. Okay, just showing the normal slide, you may not be able to visualize properly. Now let us have a look here. So only 4 structures, remember. Head, so here you can see head. From here to here, which is present is a head. And what does the head contains? Head contains a nucleus along with a Golgi apparatus. What do you mean by Golgi apparatus? Golgi apparatus, you all know, which is a part of a cell, right? So now this Golgi apparatus, which is converting as a acrosome, in the sperm. Remember the word acrosome, which is the modified Golgi apparatus. Am I clear? Am I clear to everyone? Which is a modified Golgi apparatus. So head, easy. Head, which contains 2 parts. One is a nucleus and one more is an acrosome. Acrosome is a modified Golgi apparatus. Why the acrosome is important for the cells? Why? Because acrosome, which releases the enzymes so that the sperm can easily enter into the egg. So, or the acrosomes which releases the sperms, acrosomes which releases the enzymes, and these enzymes will dilute or break the membrane of egg, or break the egg membrane. So at that time, when we are studying a fertilization, at that time it might be helpful. So keep this in a mind now itself. At that time you can tell me like, no ma'am, this is going to helpful for this. Okay. Yes. So nucleus, acrosome, done. Now there is one more structure here between the neck and the middle piece. Here one more structure is there. This is called as the neck. This is called as what? Neck of a sperm. So this neck usually contains a centrioles. Yes, we know centrioles which are present in the animal cells, not in the plant cells. So as we are talking about a human sperm, here the centrioles which were present usually in the germ cell, they come and settle in the neck of a sperm. Why they will be helpful? They will be helpful to form a fibers, spindle fibers, which makes a skeletal structure of the sperm. Okay, so it is going to produce the fiber, and this fiber will go throughout the sperm. So neck, done. Now, middle piece. Head, acrosome and nucleus, neck, which contains a centrioles, and now the middle piece. What does the middle piece contains? Middle piece contains the mitochondria, but the mitochondria in the sperm, we are not going to call that as a mitochondria. Then what do we call that? That we are going to call as a nebunkern. I'll write. That will be calling as a nebunkern. So this is what I said. What are the changes are happening from spermatid to sperm? Like Golgi apparatus is converting or forming as an acrosome, and centrioles which will synthesize the spindle fibers for the skeletal structure of a sperm, and then the mitochondria is converting as a nebunkern. Now, what is the function of this mitochondria? Mitochondria, or nebunkern, let us call it as a nebunkern, okay, because we are calling this as a nebunkern in a sperm, so let us call as it is. Okay, So this Nebenкern will helpful to provide the energy for the motility of a sperm. Yes, sperm has to move. If you can remember the female reproductive system, there sperms will be deposited in the vagina. So from the vagina, the sperms has to move to the cervix, cervix to the uterus, uterus to the fallopian tube, and especially to the ampulla of a fallopian tube. Right there, only fertilization is going to happen. The sperm has to travel a lot. For that, sperm needs the energy. Yes, because of the function. Because of its function, we are calling this as like that, and the name of the scientist. Okay, so it has to get a lot of energy for the movement. So that is why this mitochondria or the Nebenкern will helpful for the movement of it. Okay, is it clear? I hope I am. Yes. Okay, next is the tail. And what does the tail will do? What is the use of a tail in the sperm? Yes, we all know the tail will be helpful in the movement of a sperm. That's it. So this is the structure of a sperm, and let me write again. It is very difficult to get a pen from me. Okay, leave. So tell me if it is not visible properly. So head, which contains two structures. One is the acrosome and one more is a nucleus. Of course, the nuclear membrane. Nucleus. Of course, the plasma membrane will be present as it is throughout that. Okay, so what happens? Acrosome, which produces the enzymes to digest the egg membrane. Okay, function of acrosome. And the function of nucleus is to carry the genetic material. And neck, which contains the centrioles, and these centrioles will produce a filament or microtubules which will act as a skeletal structure. Okay, and middle piece which contains a Nebenкern. Nebenкern, which provides the energy, and tail, what is the use of the tail, which will be helpful for the movement. Okay, so I hope this is clear to every one of you. Let us move forward. And here you can see the significance of spermatogenesis, why the spermatogenesis has to happen. Okay, these are all the significance. And next, so this is all about a spermatogenesis. Genesis means a synthesize. What is synthesizing here? Sperm is synthesizing. So before going to the oogenesis, let us, or before going to the next step, let us have some questions. Let us discuss some questions and then we'll go. Acrosome is made up of, or acrosome is what? Mitochondria, centriole, Golgi body, ribosome. Very good. Before reading the options itself, I'm getting answers in the chat box. That's great. That's JNB Gondia. Thank you. Thank you very much. Please, everybody, answer in the chat box. Sorry, chat box or poll. Anything is fine. All others are not active. Not even in the poll. Okay, very good, very good. Now you're using the polls. That's great. Okay, I'm ending the poll. And the results here, if I say, yes, every one of you was answered in the poll and chat box, it's absolutely correct. Acrosome, which is the modified Golgi apparatus or modified Golgi body. Okay, so next question. Spermatids are transformed into a sperm by the process called what? Spermiation, implantation, insemination, spermiogenesis. Very good, very good. The so quick answers in the chat box also. I'm relaunching a poll again. Yep. That's great. No, you're getting confusion. Where comes a, okay. So I'm ending the poll and the answer here. See, few people have given it as a spermiation or implantation. Where comes the implantation in the males? No, yesterday I said implantation is the attachment of the zygote or the embryo in the female reproductive system, especially in the uterus, right? So not these three, not even the spermiation. The answer is the spermiogenesis. Okay, so hope this is clear. So now let us move to the oogenesis. Before going to the oogenesis, let us see the hormones which are helpful in this oogenesis. Okay, so as usual, like a males, in the females also, first hypothalamus which starts the production of the hormones. Okay, this hypothalamus which is called as a master gland, right? So which is going to guide our master gland, which is the pituitary gland, and the hormones which are released from the master gland or pituitary gland will guide the ovary. And we know what are the ovarian hormones, and these hormones are estrogen and progesterone. These two will be produced by the ovary, and the process will continue during the menstrual cycle. During the menstrual cycle or during the pregnancy and all. These will be seen even more clearly. Okay. So now, oogenesis. Before going to the questions which are there on the slide, oogenesis. Again, genesis means what? Synthesize. Ooh means egg or a female gamete or female gamete. Okay, so synthesis of a female gamete, this process is called as a oogenesis. Where it happens? Oogenesis will happen in the ovaries. In the ovaries. And when it starts, can anybody tell me when the oogenesis will start? The oogenesis usually, which is called as a discontinuous process. Okay, so discontinuous process, why? Because actually oogenesis will start in the, whenever the female baby starts from the birth. Yeah, you're very close, very close. Actually, whenever the baby is in the womb, at that time itself the oogenesis will start. But it is not a continuous process. It will start and it will stop for some months, and it will start and it will stop for some months. From puberty, this everything will be starts very properly. Okay, so that is why oogenesis will be called as a discontinuous process. And what is the function? As usual, to continue the next generation or to pass the genetic material to the next generation, this oogenesis is important. Okay, so I'll tell you like exactly how this oogenesis, when it starts and how it continues. First, uh, when the female is a fetus, like embryo stage, first month of embryo in the mother womb, at that time itself, the follicles, primary follicles will start. Like around 1,700 primary follicles will be produced at that time. Then in the second month, all these primary follicles starts to grow, and ultimately 6 lakhs of primary follicles will be there in the second month of embryo, female embryo. Then fifth month, 7 million primary follicles will be present, and like that. Then what happens? All these will shrink. Primary follicle starts to shrink after a fifth month, and during the birth itself, the female will be having 60,000 to 80,000 of primary follicles. Okay, during the birth itself, the female will be having a 60,000 to 80,000 primary follicles, and these primary follicles starts to grow as an egg during the puberty. Okay, so primary follicles will start from the beginning itself, but they will start to develop as a secondary oocyte at the age of puberty. Just a minute. Okay, so now let us move forward. So if you remember, if you understood the spermatogenesis, this is very easy. Now let us start our oogenesis process. As I said, in the puberty, this primary oocyte will, primary follicle will start to develop as a secondary oocyte. No, let us see. As usual, maturation. Here there are how many phases in the oogenesis? Means only 3. Not like in the spermatogenesis. Here we don't have a many phases. We are having only 3 phases in the oogenesis. One is multiplication phase. Multiplication phase. Second is the growth phase. And the third is maturation phase. Okay, so these are the 3 phases. And if you can remember the spermatogenesis, what I said there, from the germ cell it will start, right? But in the females, it will be starting from the primary follicle. Let me just explain or visualize the oogenesis in the slide only. Next slide I'll show you properly. Okay, so here what happens, a diploid primary follicle will be there, which undergoes a multiplication phase. So which gives a diploid structures again, which gives a diploid structures again, right? So what do we call these? After multiplication phase, there we have called them as a spermatogonia, right? Here we are calling them as a oogonia. Here we are calling them as a oogonia. Let me write with another color. I know this is not visible properly. Okay. So this oogonia will undergoes the growth phase next. Okay, let me show you here. So here the oogonia which form, and this oogonia will undergo the growth phase, and that growth phase will give us the structure called as a primary oocyte. Okay, and primary oocyte which undergoes the maturation phase, and that maturation phase, which means a meiosis, right? Meiosis 1 and meiosis 2. This primary oocyte undergoes the meiosis 1 and will get a secondary oocyte. There where we have got a 2 secondary spermatocyte, both are functional, but here is not like that. Primary oocyte will undergo the meiosis 1, where one will become a secondary oocyte and the one more will become a first polar body, which is a non-functional. Only the one secondary oocyte will be functional and the first polar body will not be functional. Next what happens is that this secondary oocyte will undergo meiosis 2. Only the secondary oocyte, the first polar body, as it remains unfunctional, it will remain as it is. Now the secondary oocyte will undergo the meiosis 2, and this meiosis 2 will give or will result the egg, or along with the egg, 2 cells will come again. Egg, along with the egg, there will be a secondary polar body. This is a second polar body. And this is the first polar body. So is it clear? Second polar body and a first polar body. So remember, if the sperm number must be high, that is why all this process were happening. But here what happens, once the primary oocyte has been synthesized, then, or primary oocyte has been produced, then what happens? This primary oocyte will undergo a maturation phase. Meiosis 1 occurs. Meiosis 1 will result into a 2 daughter cells. One will be the secondary oocyte. One more will be the first polar body. Now the secondary oocyte will undergo the meiosis 2, and it also results in 2 daughter cells. One will be the egg and one more will be the second polar body. Here one thing we need to remember, that usually whenever the menstrual cycle is happening, the one which is eliminated is a secondary oocyte. Okay, why not the egg? Because the process has to complete, but why only secondary oocyte is releasing? Because secondary oocyte will convert as the ootid or the egg only if sperms enters into the female reproductive system. If there is no sperm in the female reproductive system, after meiosis 1, secondary oocyte will produce and it stay as it is. Okay, is it clear? This keep in the mind. This meiosis 2 in the oogenesis will happen only when the secondary oocyte, where only when the sperms has been entered into the female reproductive system. This is about the oogenesis. Oogenesis, ultimately ootid or the egg or the ovum has been produced. Is it clear? I hope. See, if we are comparing both the spermatogenesis and oogenesis, just the step, you can see spermatogenesis has a 2 steps. One is a spermatocytosis and one more is a spermiogenesis. Spermatocytosis, again, we have a 3 steps: multiplication phase, growth phase, maturation phase. Okay, so this multiplication phase, same. There we are getting a spermatogonia, here we will get a oogonia after the multiplication phase. After the growth phase, from spermatogonia we'll get a primary spermatocyte, and from the oogonia we'll get a primary oocyte. And then from the primary oocyte, they undergoes the maturation phase. So in the maturation phase, first will be the meiosis 1. Primary spermatocyte will give what? Secondary spermatocyte. Primary oocyte will give a secondary oocyte. And then ultimately ootid, spermatid. That's it. So one more extra step is happening in the spermatogenesis. That is, the spermatid will undergo the process called as a spermiogenesis, which ultimately leads to the formation of a functional sperm. Okay. So this is it. And you can compare between the spermatogenesis and oogenesis. You can see where the spermatogenesis will take place, it takes place in the testes, but oogenesis will occur in the ovary. Okay, spermatogenesis, which has a limited growth phase, but oogenesis growth phase is elaborated. It is a kind of lengthy phase, because once the fertilization happens, the same egg has to nourish the developing embryo. That is the reason. Okay, and see, after the completion of spermatogenesis, where we are getting a 4 sperms, but after the completion of oogenesis, we'll be getting only 1 egg, right? 1 egg, and the remaining all are polar bodies. Okay, so polar bodies will be present in oogenesis, not in the spermatogenesis. Okay, so there are many other differences between spermatogenesis, oogenesis also. So I can suggest you, like, whenever you are studying both spermatogenesis and oogenesis, it is better to write both together. Spermatogenesis and oogenesis, both the process, if you are writing together, it will be very easy for us to understand and to keep in the mind. Okay. Yes. So that's it about today's class. And if you guys are having any doubts, please ask me in the chat box. Otherwise, you can answer in the poll and you can leave for the day. Thank you. Thank you all. Thank you. Okay. Bye-bye.